Insect pest management, behavioral control. It's behavioral control, pest control by sex pheromone, behavior control by trail pheromone, pest control by alarm pheromone, pest control by aggregation pheromone, mode of application, advantages, disadvantages, and which are the reference sites I used. So behavioral control, protection of resource from pest is usually achieved by poisoning the pest with a toxic pesticide, but it can also be achieved by manipulating a behavior of a plant pest. Manipulation of pestilential behavior, feeding the resources, or associated with pestilential behavior, finding the resource. Pestilential behavior finding the resource are unrelated to resource such as mating. The behavioral results from the central nervous system to various input that derive from the stimuli so that the behavior can be changed by the manipulation of input. So pest control by sex pheromones. Sex pheromones are pheromones used by an organism to attract an individual of the same species, encourage them to mate with them, or perform some other function closely related with sexual reproduction. Sex pheromones mediated pest control includes specificity, eliciting long distance response, and longevity in the field. It involves the making disruption between female producing sex pheromone and elicit responses from the males. The artificially produced sex pheromones are released in the field in small quantities to produce a cloud of pheromones. This cloud disoriented the male, so he cannot find the females. As a result, pest infestation can be prevented or reduced. BASF, artificial sex pheromone of Lepidoptera, the combined activity of two chemicals, mixture of phenethyl propionate, eugene, and geraniol, used against Japanese beetle Popilla japonica. Then pest control by trail pheromones. Trail pheromones are used by animals as navigational aids in directing their members of the colony to a distant location, varying in length from hundreds of meters in bees to meters in terrestrial insects. The sources of trail pheromones are the venom gland, Dufour's gland, and the hind tibia in Myrmicinae, and pygidial gland in Poinerinae, Pavin's gland in Dolichoderinae, and postpygial gland in Anictinae, and the hindgut in Formicinae. There is optimum dose to elicit the proper response. Trail longevity involves the volatility of compound, chemical stability, rate of diffusion, olfactory efficiency of receiver, highly specific in action. This reaction involves the treatment of synthetic trail pheromones and insecticidal bait. One is exploiting the effect of trail pheromones to attract target ants. So inclusion of subtle amounts of trail pheromones into current insecticides may result in efficient attracting and killing of the ants. Another approach is to disrupt tail formation of the target ants by dispersing large amount of synthetic trail pheromones on the ground. Trail disruption results in reduced foraging success of target ants, less competitive ability against other animals, as well as an inability to spread. Pest control by alarm pheromone. These are the chemical substances that are synthesized and released in response to predators to reduce predation risk. It causes alarm behavior, serve to rapidly disperse group of insects, usually as a response to predation. These pheromones, characteristic of social or gregarious insects, occur in some important insect pests including Apidae and Tripidae. So synthetic 4-methyl-3 heptanone, a component of alarm pheromone used by grass-cutting ants, can trap and control these insects. Pest control by aggregation pheromone. Aggregation pheromones gather conspicuous near hormone source, either by attracting them from a distance, attracting the activity, or inducing passing conspicuous to remain at the pheromone source, attract both sexes, and mediate the formation of a group of individuals for the purpose of mating, overwhelming predators, or overcoming host resistance by mass attack. The aggregation pheromone of insect, particularly the Cucleonidae, represent a powerful, sensitive tool for early detection of infestation. Monitoring and control strategies based on specifically on aggregation pheromones and their synergistic enhancement by host plant volatilities have been used extensively in management of many economically important pests. For example, grandizoic acid, a male-produced aggregation pheromone, has played an important role in the monitoring and control of the plum curculio in apple orchards. Mode of application: pheromone treatment is more appropriate and efficient in bigger fruit cultivation areas since homologous pheromone sprouts can be maintained there. An edge treatment is necessary in order to stop copulating female flying from neighboring fields. Advantages: ideal for use in inorganic fruit production. Species specific action works on the pests you need to control. No significant acute toxicity to mammals. Safe to use around beneficial insects and non-target species. Developing resistance is unlikely and it is natural to the insect's mating process. Affordable, easy to install and manage. It used properly can detect a low number of insects. Then can be used in all season. Can be used for specific exotic species. Disadvantages may be cumbersome to handle. Then not suitable for all weather conditions, unreliable indicators of pest abundance at a paddock level. Regular monitoring is still required, do not directly correlate to plant injury levels. Insect antiphedants. Definition, examples, application in insect pest management, advantages and disadvantages. Antiphedants, groups of antiphedants, advantages, disadvantages, insect attractants or insect repellents. Antiphedants. Antiphedants are chemicals that inhibit feeding in insects when applied on the foliage food without impairing their appetite and gustatory receptors or driving them away from the food. They are also called respiratory repellents, feeding deterrents and rejectants. Since donor feed on treated surface, they die due to starvation. Antiphedants are organic compounds produced by plants to repel herbivores through distaste or toxicity. These chemical compounds are typically classified as secondary metabolites in that they are not essential for the metabolism of the plant but instead confer longevity. Antiphedants exhibit a wide range of activities and chemical structures as biopesticides. Then, in addition to their role defending the plant, antiphedants often confer taste or odor, enhancing the flavor of certain plants. Examples are provided by cruciferous vegetables including mustard, cabbage, and horseradish, which release pungent oils containing glucosinolates when the plant material is chewed, cut, or otherwise damaged. The odorous compounds of garlic are thought to have evolved to deter insects. The concept of using insect antiphedants as crop protectants is attractive. Pest management in agriculture, forestry, and managed landscapes has often relied on toxic broad spectrum insecticides with negative impacts on natural enemies, pollinators, and other non target organisms. And continuous use of specific insecticides has frequently resulted in the development of resistance and in a very pest starved or for population suppression. Group of antiphedants. Triazines, organotins, carbamates, botanicals. The following antiphedants are produced from plants: pyrethrum, neem, apple factor, solanum alkaloids. Okay. Miscellaneous compounds. Compounds like copper stearate, copper resinate, mercury chloride, and phosphine are good antiphedants. Examples include rosin, which inhibits attack on trees, and many alkaloids, which are highly toxic to specific insect species such as quasinoids, extracts from quassia trees against the diamondback moth. Samadera indica also has quasinoids used for insect antiphedant uses. Mode of action: they inhibit taste receptors of the mouthparts. Advantages: affect plant feeders but safe to natural enemies. Pest not immediately killed, so natural enemies can be feed on them. So no phytotoxicity, no pollution. Potential use of antiphedants. The simplest method of using an antiphedant as a crop protectant is to apply it as a water or oil based spray in the same manner used to apply an insecticide. However, apart from neem products, there are few actual demonstrations of antiphedants efficacy in the field. John Pickett and collaborators at the IARC have shown that application of polygonal or methyl salicylate resulted in reduced aphid populations with concomitant increase in yields of winter wheat, in one case comparable to that achieved with the pyrethroid insecticide cypermethrin. Disadvantages: only chewing insects killed and not sucking insects. Not effective as sole control measure can be introduced into IPM. As the insect not immediately killed, they can move to untreated plants and damage them. New growth of plant not protected. Insect attractants. Chemicals that cause insects to make oriented movements towards their source are called insect attractants. They influence both gustatory, taste, and olfactory smell receptors. Types: pheromones, food lures, oviposition lures, poison bat. Insect repellents. Chemicals that induce avoiding oriented movements in insects away from their source are called repellents. They prevent insect damage to plants or animals by rendering them unattractive, unpalatable, or offensive. Then physical repellents: controlled stimuli repellents, auditory repellents, barrier repellents, visual repellents, feeding repellents, chemical repellents. Next is about carbamates. Carbamate pesticides are derived from carbamic acid and kill insects in a similar fashion as organophosphate insecticides. Next is about carbamates. Carbamate pesticides are derived from carbamic acid and kill insects in a similar fashion as organophosphate insecticides. They are widely used in homes, gardens, and agriculture. Like the organophosphates, their mode of action is inhibition of cholinesterase enzyme affecting nerve impulse transmission. Carbamates. Carbamate pesticides have the ability of reversibly inhibiting the enzyme acetylcholinesterase, which removes the neurotransmitter acetylcholine from the synaptic cleft, necessary for allowing intermittent signaling of messages. So, carbamates pesticides have a colorful and interesting history of discovery and development. Carbamate pesticides generally have the formula like this and are relatively polar, highly soluble in water, and chemically reactive. Some carbamates of importance are aldicarb, carbaryl, carbofuran, furan, and captan. Carbofuran, like many carbamates, is primarily used in agriculture. Its usage pattern is consistent with the general carbamate pesticide application, since the Midwest and Southeast are the two regions with the greatest need for pesticides in agriculture. Carbamates tend to hydrolyze easily, resulting in a low level of persistence in the environment, both in soil and water. Like organophosphates, carbamates are acetylcholinesterase inhibitors. Based on their chemical composition, organic pesticides are broadly classified into four types, like natural, synthetic, plant-based, mineral oils, inorganic, organic. Pyrethrum is plant-based, mineral oils, DDT, VHC, etc. are organic, organophosphate, malathion, etc. Then carbamates, carbaryl, okay, pyrethroids, Permethrin is an example. Carbamates derived from carbamic acid are organic insecticides that reversibly inactivate the enzyme acetylcholinesterase. In contrast to organophosphate inhibition, carbamate cholinesterase inhibition is species-specific, reversible, and rapidly degraded in natural environments with low pollution levels. Insecticides are derived from carbamic acid, with carbaryl being the first carbamate insecticide used in the field. Carbaryl is the most widely used insecticide due to its minimal toxicity to mammals and short environmental half-life. The insecticide carbaryl strongly suppressed the growth of all cyanobacterial species except N. punctiforme, even at a dosage of 5 g per ml. Most commonly used organophosphate and carbamate pesticides are nerve poisons and frequently involved in poisoning and suicide. These organophosphate pesticides and carbamate irreversibly inhibit acetylcholinesterase and accumulate acetylcholine at muscarinic and nicotinic synapses in the central nervous system. Then dichlorobos, diazinone, phendione, malathion, parathion, aldicarb, and digon. The most common OPs and carbamates involved in poisoning and suicide are known to cause acute pancreatitis. Pesticides: carbamates extremely toxic, LD50 8 mg per kilogram. One crystal mistaken for a seed by a bird will kill the bird. Absorbed through plant roots and distributed to green vegetation effectively banned in the US this year. Then carbofuran, somewhat toxic, widely used in lawn and gardens, extremely toxic to honeybees, somewhat soluble and tend to contaminate water supplies. Production in India caused Union Carbide disaster in Bhopal. Carbaryl. Extremely toxic, LD50, 1 mg per kilogram. Many may only be handled by professional applicators, banned for domestic use. Aldicarb. Carbaryl, one of the example of carbamates is carbaryl. Carbamates are derived from carbamic acid. They are obtained by replacing one or more hydrogen atoms by other functional groups alkyl. It was discovered by Union Carbide. He introduced it commercially in 1958. It is a white crystalline solid commonly sold under the brand name 7. Carbaryl is prepared by the direct reaction of alpha menthol with methyl isocyanate. Alpha menthol with naphthol. Alpha naphthol with methyl isocyanate. Synthesis: naphthalene is first nitrated to give one nitronaphthalene, which is then hydrogenated to the corresponding amine that upon hydrolysis give 1 or alpha naphthol. Now, insect control with bacteria and microorganisms. Entomopathogenic bacteria. Biological control methods such as bacterial entomopathogens are generally thought to be safer than chemical pesticides and offer several benefits. Their mechanism of action, for example, is typically more complex than traditional pesticides, aiming at a range of places where resistant pests are more prone to emerge. Biological control methods are bacterial entomopathogens are thought to be safer. Okay, so while entomopathic bacteria can be utilized as a standalone pest management tool, they are the best employed in rotation or in combination with insecticides to provide maximum efficacy and environmental sustainability. Enteropathogenic bacteria and chemical compounds are compatible and synergistic according to numerous studies. Other benefits include worker safety, reduced crop residues, and harvest flexibility owing to a short or no pre-harvest period, as well as the use of biopesticides in pest management program. So, enteropathogenic bacteria and chemical compounds are compatible and synergistic according to numerous studies. Other benefits include worker safety, reduced crop residues, and harvest flexibility owing to a short or pre-harvest period, as well as the use of biopesticides and pest management program. Bacilliaceae, Pseudomonaceae, Enterobacteriaceae, Streptococcaceae, and Micrococcaceae families contain the bulk of insect bacterial infections. Most of these bacteria are mild pathogens that infect insects under stress, but a small number are very virulent. The Bacilliaceae have received the most attention. Bacillus popillae cause milky illness in scarabaeus, while Bacillus sphaericus is a mosquito-borne infection. Bacillus thuringiensis is a common entomopathogenic agent that is used to control caterpillars and beetles. Bt is a bacterium that produces spores. So, sporulation is commonly linked to the formation of a proteinaceous protoxin crystal with insecticidal properties. Crystals ingested disintegrate in the stomach and are broken by host proteases to generate an active toxin known as endotoxin. This bacterium produces parasporal bodies that contain unique insecticidal endotoxins that act through ingestions via a pore forming mechanism that is harmful to the insect gut epithelium. The cryotoxins, endotoxins, are now commercially available for use against a wide range of insect pests, including Lepidoptera, Coleoptera, and Diptera species. Exposure to commercially cryopoisons used to control Lepidopteran pests has a little direct effect on non-target organisms. Applications of Bt bioinsecticides to agroecosystems and other environments often do not result in a buildup of spores in the environment, and the vitality of spores, particularly those exposed to sunshine, is shown to reduce. Bt is a microbial control treatment that is sprayed to insect infestations that cause the target insects to die quickly, typically without recycling. In comparison to other ecosystem innovations, the safety and environmental effect of EBPs should be assessed. Considering the danger to non-targeting species, plants can express the genes that encode the Bt endotoxin, making them resistant to various insect pests. According to farmer surveys, cultivating Bt crops can result in a large reduction in the use of conventional pesticides. Entomopathogenic viruses. Infect-killing viruses known as entomopathogenic viruses has emerged in recent years. Several viruses are approved for use in control of insect pests, and increased research is being undertaken to characterize and evaluate new viruses. Viruses attack and destroy a wide range of insects. EPVs are viruses that have been identified in a variety of insect orders. So, viruses can be utilized as a biological control agent because some insect pests are sensitive to viral infections. Then, insect viruses can be made up of either double-stranded or single-stranded DNA, dsDNA and ssDNA, as well as RNA, dsRNA and ssRNA. Several viruses targeted several plant pests in the global agroecosystems. A disease jaundice known as grassery jaundice, now known as nucleopolyhedrosis, was discovered in silkworms, as well as in another viral illness in honeybees, Apis mellifera. A nucleic acid is wrapped in a protein coat known as capsid in viral particles, which plays a vital role in the host cell infection process. When the virus partially enters the cell, its nucleic acid takes control of the host metabolic system, producing many copies until the cell dies. The virus is an ordinary parasite that cannot multiply in vitro. 3 insect-specific families: Bacillobiridae, Polydnaeviridae, and Ascobiridae. Viruses are extremely host-specific and non-pathogenic to beneficial insects and other non-target creatures, including mammals. The Bacillobiridae has long been regarded as a potentially ecological benign alternative to chemical pesticides. The Bacillobirus is commonly examined to discover the virus in this family that has the best chance of controlling Lepidopteran pests on crops, and it is divided into 2 categories, namely granulovirus and nucleopolyhedrovirus. The route of pathogenesis and reproduction of EPVs differ per family. However, infection is almost always transmitted by ingestion. So, the viral particle attached to receptors in the stomach and pass through epithelial cells. Infection proceeds to the hemocoel and subsequently to vital organs and tissues, notably fat bodies. Acute infections result in the death of the host in 5 to 14 days. Infected insects with bacillovirus appear white due to a major infection of the fat body, visible through a more transparent integument that thins as the illness progresses until it ruptures. Entomopathogenic fungi. Entomopathogenic fungi play a significant role in biological pest management across the world. EPF are heterotrophic, eukaryotic, unicellular or multicellular filaments microorganisms that reproduce sexually, asexually, or both, and generate a range of infectopropagules. Hypocereals, onygenates, endomopathorals, and insects belonging to the orders Lepidoptera, Coleoptera, Hemiptera, Orthoptera, Diptera, and Hymenoptera can be attacked by EPF. What is EPF? Entomopathogenic fungi. In nature, EPF produce deadly diseases and manage the population of insects and mites. They have a limited danger of targeting non-target species since they are host-specific. The fungus generation spores, Conidia and Blastospore, that infect their host by germinating on its surface and then spreading into its body via the exterior cuticle. Spore attachment to the insect cuticle, germ tube penetration of the cuticle, fungus development inside the insect body, and fungus hyphae colonization of the hemocoel are all phases in the infection process. The EPF spores are typically covered with the mucus layer of proteins and glucans, which assist in their adhesion to the insect cuticle and formation of specialized structures known as apraesoria. Then mechanical damage induced by developing mycelia inside the insect or poisons generated and released by the pathogen is the most common causes of insect death. The fungus generates hundreds of new spores on the deceased corpse after death, which spread and continue the fungus life cycle on new hosts. EPF have several biological characteristics including target selectivity, high reproducibility, quick generation time, and extended survival, all of which are important in the biocontrol of insect pests. Chemical pesticides are often employed to protect plants, and this has the reason of increasing insect resistance to numerous chemical classes, including in plant protection products. More emphasis has been made in recent years on the prospect of employing natural enemies such as endopathogens to manage insect infestation. CMs are microorganisms that kill insect pests. This might open new possibilities for controlling insect infestations. Entomopathogens are being developed for use in agricultural crops as ecologically favorable alternatives. They can be used to manage insect pests or biological control agents and increase agro sustainability. The biological control mechanism against insect pests on agricultural crops is one of the ecologically recognized methods. In the realm of insecticides, the field of microbial pesticides provide a unique opportunity to conduct prospective and predictive research.